So, couple of shop talk stuff before I jump into lecture this morning. Has anybody ordered the textbook through Connect and got it? Is it the correct book? Okay, unfortunately someone in my other class got the wrong book, so I just wanted to check. I didn't get any emails from you guys, so I wasn't sure if you guys had gotten them, how it worked out. So everyone's pretty happy with that? I hope so. $15 is a great deal. Good morning. Sign that for me. Okay, so the next thing was Connect itself. I copy my questions over from semester to semester, unless we get a new edition of the book, and then I have to pick new questions, which was last semester for this class. The book rep was with me yesterday. I ran into a problem with one of the questions. I used my son's email and signed up for some courtesy access. I'm going to go through and check. If you guys ever run into any problems in Connect, those assignments, if for some reason, when I picked the question, it was 3 different pneumonias and you had to match them up. Well, when the students went in to do the question, there were 2 pneumonias repeated. So they couldn't get it right because they didn't know which one was the one that was mislabeled. Does that make sense to you guys? In the past, you guys were able to report issues like that. They took that button away. I guess students overused it or didn't use it properly, or I don't know what the issue was. So that button is gone. I'm the one who has that button. So if you run into an issue like that, please, if you know how to, make a screenshot, take a picture, and email it to me so I know exactly what's wrong, because my courtesy access will go away. But last semester, all my students did these assignments, so I only know of one problem that I reported. I'm going to check, hopefully today, make sure that it's fixed. It's not until later in the semester. But they may have missed one, so that's how it works for Connect. If you run into an issue, right, and you're pretty sure you've looked at your notes, you've looked in your book, and you're like, I got it right. Why does it keep marking it wrong? Then definitely notify me, and if you can, take a picture of what's wrong, so that way I can send it up the line and get it fixed. Does that make sense to you guys? All right, let me see the roll sheet.
All of my Connect deadline dates are the same.
Yeah, that's my next thing. Come back around that way. So as I said, I recycle Connect assignments. So when I did it, I just quickly picked an arbitrary date. What I did yesterday is I went into all my Connects for all my classes, and I've updated the deadlines. So the deadlines for each of them match the test deadlines that you guys have. Does that make sense? So each unit has to be completed before the test, right? Ideally, you should complete them as we finish the topics, right? As we finish this topic, you should study the topic. You should take that assignment as if it's a quiz, right? Don't have your notes open. Don't have your book open. See what you actually know. Because you can do this assignment an unlimited number of times. And it's also going to tell you the answers when you're done, right? These assignments are for you to help you self-assess. I've hand-picked these questions. These relate directly to the objectives. These will definitely help you know if you're prepared for my tests or not, okay? And they're required. You have to do them. And when I checked, most of you guys had at least signed up for courtesy access. A whole bunch of you guys have licensed, which means you paid or put in your code, right, for it. So that's really good. That's the one good thing about the $15 book, that you can't get it unless you license. So I don't have to worry about people not paying for their Connect this semester. Kind of like this little maneuver. It's working pretty good. Bookstore probably hates me right now, but that's quite all right. So I don't like them either. They mark up the books too much. Right? I mean, I don't know. I don't know about any of you guys, but I wasn't rich when I was in college, so I was eating ramen noodles. So, all right, so that's Connect. The other thing is, if you go to your grades in Connect, Canvas, 2 C words, enough to drive me crazy. In Canvas, if you go to your grades, you'll see like all assignments for the semester, even though a lot of them aren't turned on. And again, you'll see old dates because I recycled. But there's a reason why I haven't turned some of them on, is because I want to make changes to them. And one of the assignments that you guys are going to do next week is called an agreement. And basically it's a syllabus quiz, right? And I like to get you guys settled in also because you're going to use a special program to do that quiz. All the assignments in Canvas are called quizzes. In Blackboard, everything was called a test, which annoyed me because some things I wanted to say quiz. Well, now I'm reversely annoyed because everything says quiz and nothing says test. Does that make sense to you guys? So, of course, in my labels, I let you know this is an exam. And you'll notice the exams are all posted for you guys right now, right? But you can't access them. 1, you need that special program. 2, you need a password from me, which you will get that day of the test in the computer lab. Me and the lab people will go around and punch in the access code for you to be able to take the test. You're going to use a program called Lockdown Browser, which will lock down the screen so that you can't get on the internet and cheat. Okay? And I use the same program for you to review your tests at home. So part of the assignment is going to be to download that or go to one of the 2 computer labs that has that program, the Hibernia Center, which I use for testing, which is in Building 1. One of my favorite places because I'm still in Building 1 as far as my office and my lab. And they're really great there and it's really quiet. Unlike sometimes upstairs in the fishbowl on the 2nd floor here, the open lab can get kind of noisy sometimes. That also has the lockdown browser. So those are places you could go and maybe do this assignment and review your tests. You cannot print my tests, right? Because otherwise then my test gets out there because you can just easily print it and share it with everybody. And then I have to make harder, more complicated tests and you won't be happy. So, it's for you, not for anyone else. Right? Make sense? Besides, if you write your own notes, right, of what you missed after the test, that's going to help you understand where you goofed and where you need to improve before the final. Okay? So other assignments are like discussion boards. We'll have another one coming up. I want to make some changes to it. That's why it's not posted yet. It does go along with this unit. Yesterday was the deadline for introduction discussion board assignment, right? Because it was twofold. You had to go to a website, do a little questionnaire, and then you had to put your results. Make sure you sign in. So that one, technically, you missed the deadline if you didn't complete it. The good news is that I have over 100 students this semester, so I'm already behind in grading. So get it done like today or tomorrow before I grade, because once I grade, not going back. Okay? So the other thing I had posted on Canvas that I wanted to show you a link to is I've done a recording that explains grading and discussion boards, how I use rubrics, how I can comment and you can comment within the grade book to me. So make sure you take a look at that. And next time I'll answer any questions in regard to that because hopefully by next time I will have graded your guys' discussion board. Okay. All right. Any other shop top questions? Anything I missed? Okay. All right. So I was recording, so if you phased out for a second, you can go back and rewind and listen to it again. And I didn't have any visuals to go along. I'm sorry. Okay. So let's get into the meat of the matter. So we're still on the topic of humans and microbes. And last time we went over the different domains. So let's actually back up a second and let's see how much you guys even remember at this point. I said back up. There we go. All right. So cell type is either prokaryotic or eukaryotic. What's the defining characteristic? What do eukaryotes have? A nucleus, which is what kind of structure within a eukaryotic cell? It's membrane bound, and we refer to things that are membrane bound like the nucleus, the mitochondria, the Golgi apparatus. What's the general term for them? Organelles, right? Notice that also on the sheet, membrane bound organelles on our chart. So who is eukaryotic? Which domain? Eukarya. Notice the matching of the name, right? That's kind of nice. And by default, bacteria and archaea are both what? Prokaryote. What do we also know about prokaryotes when it comes to cell organization? Is that organism made up of a single cell? Is it unicellular or is it multicellular? Unicellular. All prokaryotes are unicellular. So bacteria and archaea are both unicellular. Eukarya, on the other hand, mostly we think of organisms like ourselves, which are what? Multicellular, right? We are definitely multicellular. But are they single-cell eukaryotic organisms out there? Yes, and those are mainly the ones we study in this course because in that case they're microscopic, right? And this is, of course, microbiology. Cell walls containing peptidoglycan. This chemical structure that we'll go into a little bit more detail soon is unique to a single domain. It's one of the defining characteristics of which domain? Bacteria. Membrane-bound organelles, the nucleus is a membrane-bound organelle, right? Which is the only domain that has membrane-bound organelles? Eukarya. And again, that defining characteristic of them all being the eukaryotic cell type. Where can you find bacteria? Yeah, anywhere on Earth, right? Anywhere on Earth. Archaea. All environments, but they're specialists too. They tend to like which environments more? Extreme, right? So we're talking extremely hot, extremely salty, extremely cold. Yep. High concentrations of salt. Extremely salty. Where are you going to find eukarya? Eukarya. Yeah, just about anywhere that's not considered extreme, right? They don't like the really hot, the really cold, the really salty, right? So those are our three domains. Remember, those are overarching above things like kingdoms and subgroups and all the other different classification schemes you may have learned in the past. But this is for living organisms, right? Since, as I said, eukarya is a really big domain, it's enormous, we want to look at the members that are microscopic, right, that fall into the microbial world. And we want to talk about their cellular organization. Are they multicellular? Are they single-cellular? What are their cell wall compositions? Do they have a cell wall? Where are they getting their energy from? And where are you going to find these types of organisms? So again, this type of information lends real well to a table. So for algae, are they unicellular, multicellular, or both? Both, right? You have both forms when it comes to algae. You have some that are single cells and some that are multicellular. I realized this morning I didn't put a pen over there. So we got people digging through bags. This should be almost everybody. So what about fungi? Both for this one as well. Protozoa, unicellular. Anyone think of some protozoa that you've maybe looked at in biology class, or if you're in micro lab, which none of you guys are because this is the non-majors. So think back to biology. Think of any protozoa you may have looked at. Single-celled organisms. A lot of them live in what type of environments? Moist, aquatic. They swim around in pond water. Huh? Give me a description of it. Maybe someone else knows the name. Amoeba. One looks like a little foot, has cilia all over it. Paramecium. Starting to remember those cute little guys, right? Those are protozoa, right? Euglena, right? All these little things zipping around in your pond water. Do algae have cell walls? Yeah. Typically very similar to plants and made of a substance like cellulose. Do fungi have cell walls? Yes. And again, the unicellular fungi that we talk about, typically we commonly refer to those as yeast. And then the multicellular fungi are what we commonly refer to as molds. And in fact, a mushroom is a product of a mold. It is a very large fruiting body that gives rise to the next generation, right? Forms the spores and stuff. It's very large. But the mold itself is growing in the soil. Do protozoa have cell walls? No. And when you, and that's really easy to remember if you can think and remember about amoeba, right? Think about, they don't have a perfect shape, do they? They move by pushing their cytoplasm and their membrane around, right? They look like the blob. They're really fun to watch. And they're going to engulf their food as well. We might need to close the door. Can one of you guys save me the... They apparently locked the door next door. Oh, I hope ours isn't locked, but I think everyone's here at this point. Okay, so when you think about that amoeba, right, think about it wouldn't be able to make that movement. It wouldn't be able to engulf its food if it had a cell wall, right? So that's a good one to focus on to help you with this group, right, to have an image of what those organisms are like. Most of us are seeing mold, right? We don't need to visualize that one, right? And algae as well. So algae, one of their closest relatives are what? Plants, right? But those are multicellular, right? So we don't, very large multicellular a lot of times. So we don't typically study them in microbiology. But what organelle do they have that's helping them get energy, and where are they getting their energy from? Chloroplast, right? And so that's allowing them to harvest what type of energy? Sunlight, radiant energy, right? So these guys are very important to our ecosystem, right? We talked about them day one. These are the guys that are contributing with photosynthesis and producing oxygen as a byproduct, right? All the algae and the cyanobacteria in our oceans really make life possible for us on this earth. So this process, this ability is very important to us. Fungi, on the other hand, where are they getting their energy? What's their job on Earth? They're decomposers. So what are they eating? Yeah, something that was once alive, right? Or in the case of parasitic stuff, right? Stuff that's still alive, right? It's eating you now. So what is all of that? That's all organic. It was once living or is living, right? If it's a parasitic relationship, and therefore it's organic compounds. Meaning it was made by an organism, right? They're eating organisms. Could be plants, right? Could be animals that ate plants, right? It all starts with a good old photosynthesis, right? With those organisms that make life possible on Earth. So protozoa, what are they doing? Yeah, they're eating other stuff too. You know, again, imagine that paramecium or that amoeba engulfing. Amoeba love to eat yeast. So do paramecium, right? So they're eating other organisms. Now, there are some that have the ability to photosynthesize, like Euglena. So they're a really interesting group of organisms. But in addition to being able to do that, they still rely heavily on organic matter. They can do both. So as a group, mainly these are ones, again, that are eating other organisms. So where do you find algae? Yeah, we already said aquatic, right? Mostly. That doesn't mean there aren't some terrestrial ones on land, right? But where are you going to find fungi? Definitely on land, right? Mostly terrestrial. And again, there are some exceptions out there for organisms in aquatic type environments that are fungi, but they're rare. Protozoa, again, they're swimming around in the pond water, right? So definitely aquatic, but there are quite a few terrestrial ones as well. And because this course focuses on human pathogens, things that make us sick, do you think we're going to talk a lot about algae? In fact, we never will after this point. There's only I don't know if it's a species or a genus. There's a group of algae that will overgrow in our oceans. You guys know what that's called? Red tide, right? Or algal blooms. What eats that we may then ingest? More fish will eat it. There's one organism that typically in Louisiana we love, in oysters. Oysters, come on. Filter feeders, right? They're eating those algae. And those algae produce a toxin. And that toxin can become concentrated within those oysters. And then when you eat it, you can get sick from the toxin. Not because the algae is growing in your body or that it even grew in the oyster. It's that it produced the toxin, right, that you then ingest. So that's the only case where algae is bad for us, right? Can really make you very sick. Fungi we know can make us sick, right? There's yeast out there that can overgrow and create problems. Most women have experienced this, right? Our own yeast and our own vagina can overgrow under certain circumstances and be very uncomfortable. It can happen for men too, but they don't have that warm, moist vagina like we do. So it happens in other places, right? Skin folds and other issues that sometimes people have. So yeast can be a problem for us. Can fungi be a problem? Can mold be a problem? Yeah, right? There are some molds, especially after Katrina, we worried about growing in our homes, right? That could make you sick. Some people are allergic, right? That's a whole nother ball of wax, right? Allergy as opposed to illness, but still can be quite uncomfortable. Protozoa, can they make us sick? Things like amoeba and stuff. Like if you ingest natural waters that contain these types of organisms. Yeah, brain-eating amoeba, right? The ones that if you snort it up your nose, right? If you're doing a neti pot or something like that, it can cross and get into the brain. So yeah, there's definitely protozoa out there that can make you sick. A lot of them, actually, that can make you very sick if you're exposed. So those are the living ones. Then we get to non-living. So things like viruses and viroids and purons. And so we'll talk about what are they made out of. Even though they're not a living organism, they're not organized to the cellular level, they still contain components that cells have, like nucleic acid and protein. And they have particular hosts that they have to reside in order to replicate. So when we look at our microbial world, again, the domains are just for living organisms, right? So these infectious agents, these non-living things, are often their own other classification scheme. And then as we saw, we kind of broke down as far as the microbial world goes, right? Algae, protozoa. A lot of times algae and protozoa are linked together in one general group known as protista or protists. And then some microbiologists will also study helminthids, multicellular parasites, things like worms, right? Hookworm, roundworm, tapeworm. A lot of these worms that have a stage, a larval stage, and definitely an egg stage that can only be seen microscopically, right? So individuals that study these types of parasites sometimes need to have pretty skilled microscopy skills, right? Really good microscope skills. So, and even then, and I can't remember if I stole this from your guys' book or the other book, there's even more acellular agents, right, as we continue in our study, right, and I think there's some debate with this group here. But again, we'll ask the question, why aren't viruses, viroids, satellites, or purons included in the three-domain system, right? So why aren't they over on this side? What are they not? They're not living. They're not a cell, right? They're not a living organism. They're acellular. So they're non-living. They're typically referred to as agents or infectious agents. They're not organisms, although they do consist of a few molecules that we usually see in living cells, things like nucleic acid and protein. Very important, they're obligate intercellular parasites. They must get into a cell in order to use that cell's machinery, that cell's enzymes, that cell's building blocks, nucleic acids, and ribosomes to make those proteins, to make up those elements. They're what we call inactive outside of a host. Some people use the term, especially with viruses, right? We say we killed it. Well, how do you kill something that was never alive to begin with? You really can't, can you? Right? Killing refers to killing something that was once alive, right? But it makes sense to most people that it no longer can make you sick, right? The proper term is we've deactivated it, right? We've made it no longer infectious. It's inactive outside of a host. That does not mean it's no longer infectious, right? Someone can sneeze, right, in their hand, touch the doorknob, and put the flu virus right there for the next person to pick it up, right, touch your eyes, nose, or mouth, or introduce it in. That virus was outside of that body. But most of the viruses that infect us can survive in the environment. Unless, of course, you spray something like Lysol on it. Because Lysol can disrupt that outer layer, that envelope layer of that virus, and therefore stop it from being able to attach to us. We can do things that can disrupt viruses. Right? We can treat them with chemicals, right, and disrupt them so they're no longer infectious. We call this killing. So for viruses, do they have nucleic acid? Yes. Do they have DNA? Do they have RNA? Do they have both? No. This is an either or. Viruses either have DNA or RNA. Not both. Not currently, anyways. So typically, people who study viruses will even say, oh, this is a DNA virus, this is an RNA virus. Because that really does make a difference on how that virus works in our cells and how it affects us. And what that virus has to do to replicate itself. Because remember, in central dogma, we go from DNA in our cells to what? RNA, and then to protein. Well, if you're an RNA virus, how do you make copies of yourself? Because do we copy RNA in our cells? No, we copy DNA to make RNA. So those have to have their own enzymes a lot of times to be able to copy their RNA. They have to bring their own enzymes because we don't do that, right? We don't go RNA to RNA, we go DNA to RNA. And RNA is a whole nother ball of wax, right? Because RNA is typically single-stranded, right? But only it has to be a particular product in order to be read by our ribosomes. So some viruses are single-stranded RNA, some are double-stranded RNA, some are single-stranded DNA and double-stranded DNA. There's a whole big mixture of stuff. So it can get quite complicated. Which is not a virus class, so we're not going to go into all the complications. And it's not the majors microbiology course either, so we're not going to, I'm just giving you guys a little teaser to what's out there. Right? So in the core, it's got to have some type of nucleic acid. DNA, RNA, double-stranded, single-stranded, plus strand, minus strand. It's got some genetic information, right? That's going to dictate what happens when it gets into that cell. And it may have to carry with it its own enzymes as well to accomplish the goal because we don't have them, or whatever cell they get into don't have them. What helps protect this DNA? A protein coat. All viruses are nucleic acid and a protein coat, and that's what helps protect them. My numbers are going the other way. And I want it to go down, so I guess I'll go across. Okay, so viroids, in contrast, though, are just RNA. Viroids are satellites. Viroids, their definition is RNA only. Satellites, we found, could be DNA. So this is one of those gray areas of newer research, right? Where we're like, what are these things? So I've noticed that the term viroids is getting phased out in the literature, and you're seeing more satellites. The problem with that is satellites can be RNA or DNA. Purons or pyrons, no nucleic acid at all. So as I said a moment ago, viruses got to have that protein coat. At the very least, and we typically call these guys naked viruses, because some viruses will have an additional layer known as an envelope. And those, of course, are called enveloped viruses. Those are mostly the ones that infect us. And the good news with that is that that envelope can usually be disrupted by a lot of the chemicals we use to clean, like Lysol. Naked viruses, on the other hand, don't have that envelope, right? They're nucleic acid and protein. Protein's a little bit harder to disrupt than envelopes. Envelopes are very similar to membranes, right? So they've got a lot of what molecule in them if it's like a membrane. Phospholipids, right? Those lipids are very susceptible to a lot of the cleaning agents that we use. And so we can disrupt that layer. Where proteins are a little bit more stable, a little harder to disrupt. So we know we get viruses, right? Who else can get a virus other than humans? Other animals, right? Can plants? Yeah. Can fungi? If you're a cell, there's a virus out there that potentially can infect you. All living cells can be infected. And I still want to keep going down, and this thing's going across on me. Sorry. So, viroids, no protein. None for viroids or satellites that we know so far. Purons are, in fact, just infectious proteins. Right? So they're only protein. Their name actually infers that they are just a protein. So as I said, all living things for that. Viroids, probably why you haven't heard much about them, because you've ever heard anyone getting a viroid infection? No. So it's probably not human or animal, right? It is, in fact, a problem for plants, right? Where these RNAs can get in and disrupt the functionality of that cell. They interfere with what RNA is doing, right? Making proteins. Viroids or satellites, probably my favorite one, I think of these guys as hitchhikers. So the best example, a real world example to give you guys, is the hepatitis D virus. Come to find out, is not actually a virus. What it is the hepatitis B virus with the hepatitis D viroid inside of it. So we've got the hepatitis D, B, A, B, D, C, B, right? Drive me nuts. Okay. All right. So what we call the hepatitis B virus. So it's got nucleic acid. I can't remember if that one's DNA or RNA. And the protein coat. Inside of that is this extra piece of RNA that actually does something. So because of that, because it was distinctly different, they said, oh, this is hepatitis D now. Well, come to find out, it really is just hepatitis B with this extra piece of RNA. So that extra piece of RNA they call a viroid, or sometimes now a satellite. The good news with this is, what do we have to protect us against hepatitis A and hepatitis B? vaccines. And since technically hepatitis D is hepatitis B with a little extra, the hepatitis B vaccine protects you against both B and D. Getting hepatitis D or B. Still with me? I know the A's, B's, D's, C's. I'm like, oh my God. Right? So in this case, you could think of it, right? Viruses kind of have their own infectious agents, right? In this case, right? Except for those guys, it's not bad news, right? It's not bad news that something is inside of it. It's bad news for us because not only are you getting hepatitis B, but you're also getting this viroid along with it. The good news is, as I said, because it's typically mostly B with just a little extra, what we have to protect us to keep B out of our cells, hepatitis B, also keeps out hepatitis D. Okay, now that you're thoroughly B, D'd, and C'd out. Okay. Purons or pyrons, and there's even some debate on whether these really are what we say they are. But we'll stick to what's in our books for right now, and we won't get into current research. These, as far as we know, just humans and animals, right? So most of you probably heard of mad cow disease, right? That is believed to be a puron disease. It's a protein found in our brains that has a particular functionality. It can become deformed either through mutation or from ingesting an already mutated protein. So in the case of mad cow disease in England, anyone know how that whole thing got started? It got started, first of all, by probably that particular defect, right? That protein that caused other proteins to malform, damaged the brain. The brain basically looks like a sponge. It deteriorates, right? So they go cuckoo bananas because they can't function anymore and they die. The same thing happens to people, right? It can happen due to a mutation or from ingesting this. So obviously at some point this mutation happened. But why is it that they had this huge problem? I mean, what's the likelihood of all your cows mutating? Not very high, right? So how is it that all of a sudden all of them were sick? Yeah, not very smart. They thought, well, let's recycle cows. So the cows died, right? They made the cows into meal for the living cows. They were feeding cows. Yeah. They thought, why waste this, right? Small problem there, right? An infectious agent is why they died, and you just gave it to all your cows. So for a while, till they cleaned up their mess, we didn't take their beef, for sure. Right? Because the other scary thing is that if people ingested it that had been contaminated with that, you would get sick too. And we don't have any treatment for this right now. It's your own protein behaving badly, basically. How do we target that? How do we stop that? And that's the problem with viruses too, right? They get inside of our cells. You're really reliant on your immune system to detect, find, and stop that process. And a whole lot of your cells die in the process too. That's why the flu, you feel like you just want to die, right? Because your body is at war. And it's even killing itself in the process of trying to kill that virus, right? And stop it from killing more of your cells. So a friend comments to you that all bacteria should be killed to make the world safer. Which of the following would likely result if such a scenario occurred? So let's take each one of these basically as a true and false statement, right? So certain foods such as cheese could not be made easily. True or false? True, right? There are some bacteria out there that help contribute to the making of cheese, right? So we wouldn't be able to do that anymore. Humans would have fewer fungal diseases. False, right? Because do bacteria cause fungal diseases? No, fungi cause fungal diseases, right? There's a distinction, right, between disease, right, as to what causes it. No medications could be manufactured. No, that one's false, right? Because not all medications are made by bacteria, right? Some are made in a lab, some are made by fungi, right? So we could still manufacture medications. Humans would be free of disease. False, right? As we've already said, not just bacteria make us sick. Viruses do, fungi can. Plants would be free of disease. False again, right? So only thing here, right, which of the following would likely result if such a scenario occurred, right? We just couldn't make cheese as easily as we used to. But we wouldn't get rid of fungal diseases because fungi are not bacteria, right? We'd still be able to manufacture drugs, right? They're made by other things. We'd still have disease for both plants and animals and humans. All right, so that's it for our first topic, humans and microbes. So study up on that, and then once you feel comfortable, what should you do? Take your Connect assignments, see where you're at, right? And once you submit that assignment, you can do it again, right? So if you didn't score so great, study again, take it again, right? You can keep doing that until the test date. And if you are doing it more than once and you're just not getting it, obviously come see me, right? We need to maybe talk about how you're studying or what changes you might need to make to your plan, right? Because it's obviously not working if you're not getting these.